Okay, welcome back for this last section in this module on cells and cell biology. We've talked about a lot, there's a lot of information, and I've directed to your book for a good amount of reading in this module. And you'll see that as we move forward. We just can't cover everything in these videos, so we'll cover some of the more complicated stuff, and I will relegate some responsibility to you guys to do some reading around some of the subjects, which you should be doing anyway. You just delve a little bit deeper for some of these topics. So what we're going to do today is we're going to look at the nucleus and what's called the endomembrane system. And this is often a little bit confusing. So I'll go a little bit slowly here. We don't have much to cover, but we'll go a little slower. Okay, so the nucleus and the endomembrane system is a membrane-based system which includes both internal membranes and the cell membrane, the plasma membrane. So let's just look at an animal cell, and I'll leave you to look at a plant cell and draw the analogies in the plant cell. So I'm looking at over here on the left, I'm looking at this animal cell. So the endomembrane system is composed of the nuclear envelope. Okay, so that is the membrane, the double membrane around the nucleus. So the endomembrane system does not include the interior of the nucleus. It's the nuclear envelope, which is a little bit strange, but so it's the nuclear membrane, the nuclear envelope. It's then the rough and the smooth endoplasmic reticulum, the Golgi, any transport vesicles and lysosomes, and also then the cell or plasma membrane. So what is the purpose of the endomembrane system? The purpose of the endomembrane system is to chemically change proteins and lipids that are produced in the cell within the endomembrane system, and to act kind of like FedEx, in that the endomembrane system ships different things to different places within the cell and to the outside of the cell. So we'll take a look at the endomembrane system in a moment. We're going to start off by taking a look at nuclear structure before we get to the endomembrane system. So let's take a look inside the nucleus. So in the nucleus is where you obviously find the majority of the DNA inside a eukaryotic cell. And I say the majority of the DNA because there is some DNA in the mitochondria. There's a mitochondrial genome, and in plant cells, also in the chloroplasts. So we're dealing with what's called the nuclear genome contained within the nucleus. And that DNA is assembled into a structure called chromatin. And chromatin is an assembly of DNA and proteins. Now, when we look at chromosome structure in several weeks, we're going to look at that in a little bit more detail. Also within the nucleus is a structure called the nucleolus. Now, the nucleolus isn't a discrete membrane-bound structure. It's more like a region of chromatin. And that region of chromatin has some specialized transcription going on, and it's also where ribosomes actually get synthesized. So they get synthesized in two pieces and then exported out of the nucleus, and then they come together to do their job in the cytoplasm. But the nucleolus is the site of some specialized transcription, and also the site where you assemble the different proteins and RNAs into the ribosome. So that chromatin and that DNA is surrounded by what's called the nuclear envelope. The nuclear envelope is a double envelope, a double membrane. So if we're looking at just a little piece, let's just take a look. Well, let's look at this little piece down here, and we can look at a nuclear pore as well as the membrane being two membranes. So if we expand up and we can look at this, here are two pieces, and you can see the two pieces of membrane. They kind of wrap around. So it's called a double membrane because the membrane kind of wraps around and back on itself. And so we end up with this lumen, this inside space between the two membranes of the nuclear envelope. So inside the nucleus is called the matrix. That's a structure, it's a scaffold, which basically arranges the chromatin into discrete domains where you find specific chromosomes. And so then there are these little gaps in the nuclear envelope, and those gaps are actually filled with complicated protein structures that make what's called a nuclear pore. A nuclear pore is like a gateway, it's like a door. It allows things to move in and out of the nucleus. And so there's a lot of stuff happening in the nucleus, and there's a lot of stuff happening in the cytoplasm, and you don't necessarily want those two sets of stuff just mixing up kind of willy-nilly, higgledy-piggledy. You've got to control what's going in and what's coming out. And so certain proteins can go in, but they can't come out. Certain molecules can come out, but then they can't go back in again. So for example, an RNA molecule that's being made by gene transcription needs to be processed in the nucleus and then sent out of the nucleus into the cytoplasm so that it can be translated. And so that RNA leaves through the nuclear pore. But what about all the proteins you find in the nucleus? So all of the enzymes that do DNA replication, all of the enzymes that are involved in transcription, how do they get in there when they're made in the cytoplasm where translation's occurring? Well, they go in through the nuclear pore, and they have little sequences of their amino acids which tell the nuclear pore you're allowed in, and other proteins that lack that are basically kept out. So it's like a doorman, it's like a gatekeeper. Some things can go in, some things can come out, some things can go both directions. And so they have what's called a nuclear import sequence that sends them in, and the nuclear export sequence sends them back out again. So that's a little bit about the nucleus and its structure. Let's now kind of start getting into the endomembrane system. So we already saw that the nuclear envelope is part of the endomembrane system. Now the nuclear envelope is continuous with the endoplasmic reticulum. And you can see that over here on the left side of the diagram. So this is the nuclear envelope in blue. The nuclear matrix is inside the nucleus. So the blue is the nuclear envelope there. So you see how this membrane kind of wraps around, and then it actually becomes the membrane of the endoplasmic reticulum. And so we say that the endoplasmic reticulum is contiguous, which is just a fancy word for continuous, with the nuclear envelope. So really, in terms of space, the lumen of the nuclear envelope is the same space as the inside or the lumen of the endoplasmic reticulum. Now there are mechanisms to make sure that certain things happen in the nuclear envelope and other things happen in the smooth ER and other things happen in the rough ER. So that's the next point to be made. There are two types of endoplasmic reticulum. There's rough, which tends to be closer to the nucleus, and then there's smooth endoplasmic reticulum. So this word endoplasmic reticulum, endoplasmic means, endo means inside or within, plasmic refers to a membrane structure, and reticulum comes from the Latin meaning little network. So this is a network of membranes inside the cell, and it's usually close to the nuclear envelope and extends away from the nuclear envelope. So you can see that the endoplasmic reticulum is kind of made up of these stacks or these plates of membrane that then wrap around the nucleus. You must think about this in 3 dimensions because the artwork shows it reasonably well in 3 dimensions, but you've got to think about your nucleus as a ball in the middle, let's say in the middle of the cell. That's not necessarily true, but in the middle of the cell, and then you've got these sheets of membrane around it which are all connected. Close to the nucleus we've got rough ER, and then further away is smooth ER. So what is going on in this endoplasmic reticulum? Well, let's talk about rough versus smooth. Rough ER is rough because it's covered in ribosomes. So I said earlier in this module that you find ribosomes and translation occurring in the cytoplasm. So those are free ribosomes in the cytoplasm. That is where all translation begins, except in the mitochondria and chloroplast. But all translation initiates in the cytoplasm. Some of those ribosomes are actually then sent to the rough ER. So the ribosome knows to take certain proteins as they're being synthesized to the rough ER, and the ribosome then docks on the surface of the rough ER, and at that point the ribosome carries on translating its protein either into the inside of the endoplasmic reticulum, or it inserts the protein directly into the membrane, so the protein can span the membrane with a part in the cytoplasm, a part inside the endoplasmic reticulum, and then a short region that spans the actual membrane. So the rough ER is rough because it's covered in ribosomes that are engaged in translation. So that's one of the main functions of the rough ER. What about the smooth ER? The smooth ER does a lot of biochemistry. For example, it stores calcium, and that's going to become super important when we talk about cell signaling. It's also involved in detoxification. So if you look in cells in your liver, if you've got somebody who drinks a lot of alcohol or is taking a lot of toxic drugs, you'll find a lot of smooth endoplasmic reticulum. And one of the reasons that your physician will tell you you shouldn't have alcohol with certain drugs is that most drugs are to some extent toxic. And if you're consuming alcohol, what happens is the amount of smooth ER in some of your cells goes up, and those are the cells which are detoxifying certain compounds. So if you crank up the amount of smooth ER in your cells, you're basically detoxifying things like antibiotics and things like that, because they are ultimately foreign to your body, and so they're being broken down, and some of that happens in the smooth ER. So alcohol can increase the amount of smooth ER, and that increases the breakdowns of some drugs, and so some drugs you're told not to consume with alcohol. So detoxification is important there. You also find lipid synthesis occurring in the smooth ER. So we've talked a lot about membranes. We've not looked at the structure, but you know at this point that membranes are made of phospholipids, and they've got fatty acid tails. So you know membranes are made of primarily lipids with a lot of protein, but primarily lipids. And so where is all this membrane coming from? How do we get all these lipids? They don't just kind of Harry Potter out of nowhere. So a lot of those lipids that are used subsequently to build things like phospholipids are made in the smooth ER. And once those lipids are made in the smooth ER, they've got to get to all the various places they're needed. Maybe they're needed back to synthesize a new piece of nuclear envelope if the cell is dividing and we now need 2 nuclei. Maybe the cell is growing and we need more plasma membrane. And so in that case, we've got to make more lipids to make more cell membrane. So that's an important function of the smooth ER. In both of these compartments, the rough ER and the smooth ER, you also find a process called glycosylation. So glycosylation simply means adding sugars. Now we talked about carbohydrates, and I kind of kept carbohydrates and proteins and lipids and nucleic acids separate, but let's complicate it a little bit to reflect the actual complication that you do see and the complexity you do see inside cells. You can take a lipid, for example a lipid that's being made in the smooth ER, and you can chemically add a sugar to it. And you can actually add these long chains, branching chains of sugars. And you can do the same thing to proteins. So in the ER, you also see protein glycosylation and lipid glycosylation. And what is the point in that? The point in that is that some of these proteins and lipids will end up on the cell surface. And those sugars act as important signals to other cells. They also send information internally so that certain proteins and lipids know where to go. So variety of roles of glycosylation, and that can start happening at least in the endoplasmic reticulum. Let's just take a look at what this would look like in an actual cell. So this is an electron micrograph. This is a transmission electron micrograph. We've not seen any of these kinds of images before. Now I don't want you to walk away from this thinking, wow, look, the endoplasmic reticulum is bright green and the nucleus, which is up here in the top left, is blue, and this down here is actually the edge of a mitochondrion, and mitochondria are purple. This has all been added on. It's all pseudo-colored just so that we're not looking at a dull old black and white image here. So this is some rough endoplasmic reticulum, and you can see it's this kind of network of membranes and these filaments. And you can see, look, this piece here is actually the same as this piece here because it's connected in the middle here. And you got to think about this in 3 dimensions. If you think about this wrapping around the nucleus, there are going to be lots of these places where the endoplasmic reticulum, different parts of it are connected to one another. So it's often the same compartment, but there are strategies for actually biochemically doing discrete things in rough versus smooth. So what about all those red dots? The red dots are ribosomes, and they're going to be docked on the surface, translating proteins into the lumen of the ER or into the membrane of the ER. So that just gives you a little bit of an idea of the structure that's not being created by an artist. So that's what it would look like if we froze a cell and then took a section and then looked at it with a very powerful microscope. So that's an incredibly powerful microscope using electrons rather than light. Okay, so let's now move on and talk about the next part of the endomembrane system, the Golgi, named after a famous Italian scientist. The Golgi is gonna, in some cells there'll be multiple Golgi, and they're often moved away from the endoplasmic reticulum, so between the endoplasmic reticulum and the cell membrane. So let's do the function first before we look at the structure. What's the function of the Golgi? What is the point of the Golgi? The Golgi is very much like, it's like a post office, I guess. material comes into the post office. So I ship a parcel, it goes into the post office, and then the post office sends it back out again. So the Golgi apparatus is kind of sorting and processing material that arrives. So where is that material arriving from? Where's it going to? Well, it's arriving from the endoplasmic reticulum, and it's going to a whole number of other places in the cell. So sometimes things will be going to the cytoplasm, sometimes they'll be actually being shipped outside the cell. So we'll talk about within the cell in a moment, but let's think about what would happen to, let's just think about what would happen to a molecule of insulin. So you're probably familiar with insulin. Insulin is it's a small protein, not very many amino acids, and it's translated initially in the cytoplasm, but that ribosome goes to the rough ER. And so the ribosome docks on the rough ER and then translates the insulin molecule inside the endoplasmic reticulum. So in certain cells in your body, in cells called the islets or Langerhans, you have insulin being produced at very high levels. So the insulin accumulates inside the ER lumen, inside the ER, and then as it moves through the ER, it arrives at kind of this outer face of the ER. And on the outer face, little pieces of membrane can pinch off. So it's very much kind of like grabbing a handful of membrane. So if you think about a membrane, it just kind of pinches off. And you've got a little ball of membrane. And inside that little ball of membrane, you have a whole bunch of insulin molecules. And then the insulin molecules, for example, would move in this vesicle through the cytoplasm. And here's something interesting. A lot of those vesicles, these little packets of membrane, actually move along along microtubules. And we'll talk about motor proteins and microtubules in a different video. But these proteins are moving along, these vesicles, are moving along microtubules on little motors, like little scooters. And so the vesicle comes to this face of the Golgi, which is called the cis face. And so there are three parts: the cis, the medial, meaning the middle, and then the trans, which is the face that is towards the plasma membrane. So the vesicles kind of dock and they bud into and release their contents into this cis side of the Golgi. So you can think about the cargo inside the vesicle, and when it fuses with the Golgi, the contents of the vesicle are spilled into the Golgi. And so in the Golgi, again, there's a lot of glycosylation of these proteins. And so our insulin molecules could move through the Golgi, and so they bud off and they move through and they bud off and they move through until eventually they get to this end of the Golgi. And then they bud. And in the case of insulin, the vesicles sit waiting in the cytoplasm, and then a signal from outside basically tells the vesicle, release your contents. And so the vesicle kind of spills its contents outside the cell. And in the case of insulin, the molecule's then taken into the bloodstream and distributed throughout the body, where it then modulates glucose uptake into other cells. So the Golgi is basically a processing center. So that's how something would get from the Golgi, from the ER, to outside of the cell. Now, some molecules are not destined to leave the cell. Some molecules are destined to have functions within the vesicle inside the cytoplasm. And so some of these vesicles undergo maturation processes to become structures called lysosomes. So lysosomes are vesicles which contain enzymes called acid hydrolases. And these enzymes basically hydrolyze molecules that the cell needs to break down. Maybe these molecules have been brought into the cell and they need to be degraded into their constituent components, and so they need to be released into the cytoplasm. So these lysosomes basically help to break down material that the cell no longer needs or wants to just recycle. So as what I've got here is basically two parts of a molecule that have been brought into the cell that's found its way into the lysosome. So remember, R just means part of the molecule, the rest of the molecule. So there are two parts to the molecule, R1 and R2, and this is a covalent bond. And the acid hydrolase is going to use water to break this bond between two parts of the molecule. So this could be an amino acid, and this could be an amino acid, and we're going to use an acid hydrolase to break or hydrolyze that bond using a water molecule. So that gives us an amino acid with a hydroxyl group then, and an amino acid with just a hydrogen attached to it, and we've done hydrolysis. So this is the application of hydrolysis. These are called acid hydrolases because they work at very low pH. Okay, if you put these enzymes in the cytoplasm, they don't work. They've got to be in this lysosome. So what happens is when the lysosome forms, when the vesicle comes off of the Golgi and ends up in the cytoplasm, in the membrane of that vesicle, there are these pumps, there are proteins which force protons into the vesicle. And as the proton concentration goes up in the vesicle, what happens to the pH? That's it, goes down, remember, so proton concentration up, pH down. And when these acid hydrolases are in the right pH, they can use water to break molecules down. Now, again, in this section, I've not covered everything. I can't. This is just to give you a framework. So you must focus on the learning outcomes. There's going to be a bunch of stuff in there that I've not talked about. Get some discussions going if you need help on that. Plenty of stuff in your book. Plenty of other videos in here about motor proteins and things like that. So get into those and see what you can find out. And let me know if you have any questions. And I'll see you next time.